Welcome, everyone. Thank you for joining us for today's session, Pioneering Our Understanding of the Human Brain. It is my honor to introduce to you Dr. John Donoghue, Founding Director of the Wyss Center for Bio and Neuroengineering. Dr. Donoghue is one of the most recognized researchers in the field of neuroengineering and a founding father of the merger between neuroscience and cybernetics. His BrainGate neurotechnology that aims to restore movement to people with paralysis has been recognized internationally and won multiple awards. For a complete biography on Dr. Donoghue, please look at the Biography tab at the top of your screen. Dr. Donoghue will be presenting on merging minds and machines, brain-computer interfaces to restore movement and communication for people with paralysis. My name is Dr. Roshana Kashemian, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. We'd like to extend our special thanks to the Brain Initiative Program Directors, and in particular, Dr. James Natt, for their efforts in organizing today's session. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting your questions during the presentation. Simply click on the Ask a Question box located at the far left of your screen and type your questions in the drop-down box. Our speaker will respond to your questions via email following the presentation. The presentation is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right corner of the presentation window and follow the process to obtain your credits. Now, without any further ado, please join me in welcoming Dr. John Donoghue.
Thank you for the introduction. This is Matt Nagel. He has a spinal cord injury, and he's unable to move his arms and his legs. But in fact, he is playing this video game with a chip or a microelectrode array in his brain that allows him to think and move the cursor on the screen. So what I'm going to tell you about is a brain-computer interfaces that started with Matt in 2004, and over the past 15 years there's been great advances in how these brain-computer interfaces work. And what I'm going to do is tell you about their progress and also about some of the big challenges we face in making a device that can work to help paralyzed people. There's a large community of paralyzed people, ranging from the lady you see at the top, who's completely locked in with Lou Gehrig's disease or ALS. This is a video of her, she can't move at all, ranging to a larger number of people who've had a stroke who cannot use their arm at all or not use it well in everyday life. So brain-computer interfaces aim to help these people communicate and move again. So let me start by showing you the vision of what we want. This is looks like a typical person drinking a glass of water, but in fact this is our vision of how we would like a brain-computer interface to work. We have a small electrode array implanted in the arm area of the motor cortex that picks up thoughts about wanting to move, and this is transmitted to a radio and signal processor under the skin that can take the signals and radio them outside to a small computer-like device worn on the belt, and that device can decode the patterns of brain activity, and then that can be shipped or transmitted to a stimulator that goes to wires that connect to arm muscles through the nerves, and effectively taking the brain signals and using them to control the arm muscles. So we've effectively made a brain-computer to arm interface that is made out of physical connections rather than the usual biological ones that connect our brains to our bodies. So this is our vision. So I'd like to talk about the complexity of actually realizing such a dream. So first, one of the really big challenges is that brain-computer interfaces require the interaction of neuroscience, computational science, engineering, and clinical teams all working together. The components of building this brain-computer interface are really also challenging. It starts with a neural interface that picks up signals by being implanted or on top of the brain, an implantable processor that is used to amplify the small electrical signals and do some initial signal processing, ideally inside the body, and then that's radioed out to some kind of computer that can take that data and decode those brain signals into command and control signals using sophisticated algorithms. We then transmit those signals to a variety of devices that can be useful to people who are paralyzed, such as a computer for communication, a robotic arm that's kind of a surrogate arm for people who are paralyzed, or to their own arm or other parts of their body like their legs to restore movement. So here's the system as it existed in 2004 and also as it is still used today. It starts with a small chip of 100 microelectrodes that is placed in the arm area of motor cortex where the signals are for arm movement. Today's version of the system has a plug on the head where all of those wires and leads come out. They're processed, and I'll represent the pattern of neural activity as a QR code where the black and white dots represent individual neuronal activity. And just imagine this is the activity of a patch of brain, and our job is to decode that pattern of activity and turn it into something that can be move a cursor on a screen, and I'll explain more about how that can work in a minute. So since the last 15 years, we've shown that people with spinal cord injury, stroke, or ALS all can use signals from their motor cortex to control computer cursors. There's 3 examples here. There's been more than 12 people involved in what we call the BrainGate study. It's the device that we've been developing, the system we've been developing. And from those people, we have more than 25 years of experience in how these systems work. So one of the big advances, and I'll show you 3, is the use of the system to spell. So this is a person with ALS, and she's using the neural output to copy that sentence at the top, when the sunlight strikes, and she's, you can see very rapidly pointing and clicking. And Krishna Shenoy's group, working with the BrainGate group in our clinical trial, has shown that people can type about 30 characters per minute. You and I can type about 90 characters per minute. So it's not as good as you or me, but it's still very fast communication for someone who can't use their hands or speak. Even more impressive, or a next big step, is the ability from that same tiny patch of the brain to get signals that can control something as complicated as a robotic arm that has to move in 3-dimensional space and grasp. And here you see one of our patients, Kathy Hutchinson, who had a stroke, and she's drinking for the first time in the 10 years since her stroke. She's able to lift up her morning coffee and bring it to her mouth and serve herself rather than having someone else serve her the drink. Now, even closer to the idea of a person controlling their own body is Bill here, who he is a spinal cord injured person. He's unable to move except to shrug his shoulders, and he has actually 2 of these electrode arrays in his brain in the motor area, and they're being fed to stimulating electrodes that are put into the muscles of his arm. So his brain signals are being used to control his own arm, and you'll see him able to use his arm and his hand to reach out and grab his morning coffee cup. So you can see in each of these cases of controlling a cursor to type, to controlling a robot arm, or even the person's own arm, they're really an impressive advance for people who are otherwise completely paralyzed. But they're not as good as a typical person. And you can see by the wires and connections that these people have, this is not a system that's usable at home at the current time. It's still a research-level device. So there are a number of challenges that are left that are really big challenges to overcome in order to make this a human-ready device. So some of them are in the area of neuroscience and computation, in engineering, and also in the clinical domain. So what I'm going to tell you about now is how we've chosen the area, the problems with decoding, the neural interface development that's ongoing to improve the quality of the device, and the final steps towards making a practical system for everyday use for humans. So one is what area we choose, and many of you are familiar with the famous homunculus. which suggests that if you wanted to get signals out from the motor cortex to control an arm, you'd need to have electrodes over a many centimeter square area of the brain. But this map, thanks to the work that's been done actually with humans and non-human primates, shows that in fact the organization is not quite this way, and we have to actually go back to Penfield and Jasper in 1954, who made the original map but warned us that this is actually not how things are organized. In fact, now when we've looked at higher resolution, we can see that that tiny patch where we're recording actually has a much more complex code for the entire arm inside each little area of the motor cortex, like that QR code I used as an analogy. So in fact, if it weren't true that there were mixed arm and hand, shoulder and elbow information in that small region, then Bill wouldn't have been able to reach and grasp his coffee cup. But let me just show you an experiment that makes that organizational advance a little clearer. So this is an experiment done by Carlos Vargas Irwin in our group, and it's really, I think, an elegant experiment in which he showed in monkeys that he could reconstruct the entire arm's movement from one small patch of the motor cortex. So here, what he did is he trained a monkey to reach and grasp out to toys that were swinging on a string, so a cube, a ball, other different shaped objects. And he recorded from the brain while the monkey made these reaches and grasps, and he also recorded the monkey's arm movement. So what he was able to do is he was able to show a movie that showed you exactly what the monkey's arm was doing, and that's the ghosted image. And then he used the neural data from that small patch to reconstruct what the arm could do. And what you can see is that all of the arm in the solid part of the movie, the solid reconstruction, is moving. Fingers, wrist, elbow, shoulder are moving. And although it's not a perfect reconstruction, the reconstruction is pretty close to the real thing. That is, if it was perfect, both of the shadowed image and the solid image would be together. And they're a little bit off. So what this demonstrates is that we can actually, from a small sample in the cortex, reconstruct a lot of what the arm is doing. Why we can't reconstruct it completely accurately remains a mystery, of something that we need to investigate deeper in basic neuroscience research. Now, let me give you just an intuition of how we can turn those patterns into predicted movements of a cursor for a paralyzed person. So what we do is we show a patient that has had an electrode array implanted a movie of a cursor moving on a screen. So the movie that you see is a cursor that a computer is generating. You can see the cursors going left or right. And what we do is we say to the person, imagine you're moving a mouse that's moving that cursor. And when they do that, that generates patterns of activity in their brain. So the pattern on the top might be generated with a rightward thought of movement, and the leftward movement, imagined movement, would give the bottom pattern of activity. And what we do then is we store those patterns in a computer, and then we say to the computer in the future, when you see that pattern, move the cursor in that direction. Top pattern, move it right. Bottom pattern, move it left. And you remember from the earlier slide that I showed a cursor moving on a screen, and of course it isn't perfect like the animation from the computer. You can see that it's irregular, a little bit slower than normal. And the reason for that, again, is that we have an incomplete sample. Those black squares on top of the QR code show you that we have really an impoverished or an incomplete sample. So we need either algorithms that can make up for the missing information, or we need better samples. So one of the ways that we can get better samples is get better electrodes. And the electrode that's been used in every single human study so far is this electrode array that's been developed by Dick Norman, that's placed by a surgeon into the motor cortex, and it leads out to the outside via the plug on the head. Now, these electrodes are really a marvel of engineering, but in fact, they have some issues. So you'll see two. One is that the material on them degrades so that they don't last as long as we would like, and they only get one sample of neural activity per penetration, per electrode. So first problem, you can see that these pictures of electrode tips on the left is what it should look like. That's a control electrode that's never been used, and that there's a nice plastic insulation to the tip, which is required for the electrode. But after a year in the brain, that insulation begins to degrade because the body attacks it. And when it degrades, the signal quality goes down. So here we see just a graph that shows Z is the impedance of the electrode, a measurement of leakage, and amplitude, AMP, is the size of the signal. And you can see both of those decrease over time. But if you look at the time interval, it's over years. And when the lines intersect with the dashed lines, that's when the electrode is expected to be completely ineffective. So these data were taken from original experiments in monkeys and based on a lot of observations, over 1,000, showing that, in fact, we can get very important data on the lifetime of these electrodes and that we can show they last for many years. And, in fact, the electrode that was in Kathy's brain, where she controlled that robotic arm to drink her coffee, had been in place for 5 years and was still functional. So we know we can make electrode arrays that last for many years, but, of course, we really need them to last a lifetime so they can always be functional. So there have been a number of innovations recently, although there's still a big transition phase to go from animal work to humans. But one of them that is most exciting to me is this electrode that's being developed by IMEC, by Tim Harris, Barun Duda, and a number of other collaborators. This electrode has on it more than 960 sites. So remember, each probe on the electrodes we use now has one. These probes have 960. The digitizer can record 384. So if we put several of these electrodes in, we could record not 100 neurons, but 10,000 or more neurons. The only problem is that this device is, as you can see on the left side, is rather large. It sticks up out of the skin, and so it'll have to be miniaturized more and have electronics. And the other problem is that if we have thousands of channels coming out, the data rate is higher than anyone can handle. A radio can't broadcast that much information. The power demands are huge, and we don't know the safety profile. So those are all things that have to be overcome to make an electrode that can work all the time in a human with paralysis. Now, lastly, the practical human system is going to require something that can be used at home, allow the person to be mobile. And you can see Kathy sitting in her wheelchair here, and the yellow arrows point out how there's a big cart of electronics. There's a technician in the room. And the plug on her head has a big amplifier that's attached to it in order to amplify these signals. And so these are things that have to be overcome to make a practical everyday system. So one of the very important steps forward is to be able to implant the electronics inside the body. So here's Matt with his device attached, just to give you a sense of the size of this attached electronics. And here's what we're aiming to do. It's from the movie, and you can see this small flat object. This would be implanted inside the head. And this is something that I've been working with my colleagues at the Wyss Center in Geneva, which is a Translational Neurotechnologies center, using industrial processes funded very generously by Hans-Jörg Wyss and his philanthropy to make this 100-channel amplifier that can actually transmit more than 100 channels at very high bandwidth out of the body. So this is a major breakthrough. It's going to take a few years to be realized, but it's coming now. So the translation to humans is really a very complex and challenging process. As I said, it takes the integration of many disciplines. We need a neural interface that will allow us to sample large numbers of neurons and will be long-lasting, will last in the brain for a lifetime. We need implantable processors, and the implantable processors, as I said, are coming soon. We hope to have them available for humans in 2 or 3 years. We have to decode this information, and we can admit that the kind of movements, either cursor movement or robotic movements, are not typical of everyday movements. So we need better understanding of the code in the brain in order for us to be able to produce realistic, naturalistic movements. We have a number of useful devices to connect those to, and I think we can be very encouraged that we can connect to computers or robotic arms or hands or even back to the body. And there's even a group trying to attach the brain signals to the spinal cord to restore locomotion. So the future is very exciting. Now, lastly, the Neurotech, we can see, has the potential for many, many benefits for people with paralysis and people with other disorders as well. We can see that restoration of function is possible, although it has room for improvement. We can eventually create independence, so people who are paralyzed are not dependent on others for everyday life. And since they are independent, we can reduce healthcare costs so that they are not so dependent on a healthcare system and very expensive care. And finally, one of the things that's very important to consider when we start recording from or stimulating the human brain is the ethical considerations. Of course, we need to make sure that these kinds of devices are accessible to all. They're very expensive devices, and we have to find ways to make them affordable. We are now tapping into the brain, so there's a privacy issue. Are we reading their minds? How do we protect people from not having their thoughts broadcast to others? Can we, in fact, use these kinds of systems to evolve kinds of mind control, things we have to think about. We also have to be careful that people don't make claims about what can be done with these devices that are not true. This is certainly a misrepresentation of what you can do is an ethical challenge. And lastly, a challenge that will come in the future is augmentation. What happens if we can alter the body and provide functions that we ordinarily can't have? So with that, I'd like to say that all of this effort requires a very large team. My colleagues at Brown, at the VA Hospital, Mass General, Case Western, Stanford, and at the Wyss Center are all part of a very large team who have been working together and have made all of this effort possible. So with that, I thank you very much for your time.
Thank you, Dr. Donoghue, for that excellent and engaging talk. Please remember that our speaker will follow up any questions about his presentation via email. Coming up next, join our first presentation in the clinical updates track as we hear from Dr. Wolf Julian Neumann, who will present Towards Artificial Intelligence-Based Adaptive Deep Brain Stimulation in Movement Disorders. Please join us there.